Good morning, all. Today, in the event of the International Young Scientist Awards, I'm going to deliver the talk on differential gene expressions of the gene and transcription factors for drought stress in groundnut. Mainly, in the groundnut, the major issues is the drought and the salinity. And there are several regulatory mechanisms which may regulate while you are imposing the stress. I can say the gene transcription factors, which is very essential, can be regulate while we are imposing the stress. Now, the introduction says that the groundnut, the chromosome number is a 40 and the genome size is 2.7 GB. It's mainly grown in the tropicals and the subtropical regions of the world, especially in the India, and the next one is China, which is the large producers. As we know, the genus is Arachis, and mainly it depends on the leaves and the flowers how it goes. So the productions and productivity it depends on the leaves and the flower itself. If you see the proximate compositions of this groundnut itself, the maximum fat person is like the 48 grams and the energy level is 570 kilocalorie. If you see the productions data, in the productions data, I can say that the India, which consisting of the 7 metric tons, apart from that, the productivity is 1750. The similarly way, the Gujarat is in the top positions in which it gives 3 metric tons of the productions along with the productivity of the 2670 metric millions per hectares. Now, before proceeding with that in the abiotic stress, first of all, we should know that the abiotic stress is defined as the negative impacts of non-living factors on the living organism in a specific environment. A prolonged periods of the normal low rainfalls leading to the shortage of the waters. Moving ahead with this, another things which is very important related to stress, that is the drought conditions. Now in the drought conditions, mainly the interference with the photosynthesis alter the water retentions, respirations change membrane permeability, and the interference with the nutrients mobility is the key features while you are imposing the drought stress. Moving ahead with this, there are the four critical stages in the groundnut for the productions in the productivity point of view: the emergence, flowering, pegging, and the port fillings. So those are the days which is very key important in regards to the productivities of the plant itself. Moving ahead with the transcription factors. So the transcription factors, I can say the specific transcription factors may be a regulate while you are imposing the stress. So that one is a non-transcription factors, which is contain the controls in the rate of the transcription factors for the genetic informations from the DNA to the messenger RNA by binding to a specific DNA sequence and help to regulate the expressions of the gene itself. Moving ahead with this, the another thing which is very important, that is the NAC transcription factor. So NAC transcription factor, it has a different domains. The first one is the C-terminal regions, another one is the N-terminal region. So those are the N-terminal regions, they are not variable, but the C-terminal regions are the variable. So those are the C-terminal regions which is essential for the regulations while you are imposing the abscisic acid stress. So when the drought is occurs during that course of period, there are the two regions which is called the C-terminal regions, which may regulate in response to the abiotic stress, like the ABA regulation. Now in the ABA regulations, the ABA accumulations may be occurs when we are providing the stress. The largest plant-specific transcription factors family that regulate the response to various abiotic stress, like the drought tolerance. It's not only restricted with the, I can say, salinity or the drought, it also reflects in the heat stress also. Moving ahead, as I told you that it has a different domain. The one is a nuclear localization, another one is DNA binding. Apart from that, the DNA also form a homodimers and the heterodimers with the other domains containing proteins. So it's highly conserved, and I can say more than 500, 150 to 60 amino acids which are regulated. As I told you, the highly divergence regions is the C-terminal region, which is responsible for the diversity in the regulations of the transcription activations of NARC protein, which is a functional domain and operate as a transcriptional activator or the replacer. It also possesses protein binding abilities. Moving ahead with this, another thing which is important is the mechanisms. As I told you that ABA dependent and ABA independent mechanisms are the key features for the regulations of the NARC transcription factors. Now in that case, what happened that we have to emphasize on the different research aspects. The first one, already we took the cultivated variety, which is the susceptible one, and another one is a I can say drought tolerance, that is a wild cultivar Arachidiurensis. So the objectives which consisting of study the physicochemical changes in the GJG22 and the wild cultivars during the drought stress to retrieve the conserved to retrieve the conservations, I can say, of the gene transcription factors, genome sequence of the Arachidiurensis in silico methods, and to identify the differential gene expressions of the retrieved gene transcription factors, which is to be very important. Moving ahead with this, the another thing which is very important in the event, I can say, is the experimental material. So in the experimental materials, I can say the one thing you should know, the one is the wild cultivars, those are the Arachis turanensis, which is a wild herb, right? So in this case, the drought tolerance or the wild type Arachis is procured from the directors of the groundnut research, and another variety which is very essential in the experiment, which I can say GJG22, which is cultivated mainly in, I can say, in the area of uh, 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 western belts of India, specifically in the Gujarat regions. The main features, which I can say, if I'm not wrong, it has a higher oil contents and the tolerant to the color. If you see that in the Umbrella or in the belt of the Saurashtra regions, the major issues with the collar road disease, which can decline the productions in the groundnut cultivations. So it is a mandatory that we should do certain kinds of research in relations to the wild ones and the improved one. But the main features, or I can say the drawbacks, which consisting of doing this experiment is to dig out the certain transcription factors which is responsible for the traits. If you see this, the conditions in which, as I told you, that the conditions in which the first one is the controlled ones, and another one is, I can say, the imposing the drought conditions, and the last one, which, if I'm not wrong, it's the conditions in which well water conditions. So if you see the figures, and the figures itself says then the treatments are different treatments, the variety ones, that is a controlled one, Gujarat groundnut 22 numbers, which impose the distincts or the different variations. The similarly way, if you see that in the another figures, it shows as a distance difference under the different treatments, either it is in the control conditions or the stress conditions or the revived conditions. Moving ahead with this, another thing which is important that the biochemicals or the physiobiochemical analysis. The five things which is to be important in which to dig out the really possible cause of the drought stress, that is the relative water content, electrolyte leakage, chlorophyll content, osmotic potentials, and the proteins. So these are the components which are the key features when you are regulating the drought stress. So in the conditions of the relative water content, those are the wild cultivars. Again, I'm saying those are the wild cultivars. The inclines of the water content is as compared to the GJG22 is it's major things when you are imposing the drought. The similarly way, if you see that the electrolyte leakage when the ABA accumulations and down regulations parts, it increase in the susceptible ones. Moving ahead with this, another thing which is key features which you can say that the chlorophyll content. So the wild cultivars, if you see that you don't need to give the irrigations and the timings like the 3 to 4 irrigations need to be required. But when you're providing a list of the irrigations, the percentage of the chlorophyll also is stable in the conditions of the wild at the summer seasons. The similarly way osmotic potentials, proline accumulations, and another thing which is the transcription factors. So mainly those are the transcription factors which I can say regulating which required in silico methods, and we decided that the specific size and the fragments need to be required at the particular melting temperatures. So after the RNA isolations, if you see that the stress conditions or I can say the revival conditions or the control conditions distinguished can be visualized on the gel electrophoresis. Moving ahead with this, the quantification, it's not the big deal, but the screening is important. So if you see the amplifications of the bending patterns of the duranensis in which the amplifications betting or the fold change are increased or the high as compared to the GJG22. Moving ahead with this, another things which is I can say the fold change and the CT value. So the cycle times need to be required for the multiplications in the initial stage in which the difference is to be found when you are using the endogenous control. So in the endogenous control, the tubulin, glyceraldehyde 3-phosphate dehydrogenase, and the alcohol dehydrogenase are the three endogenous controls which may be a reflect or I can say a regulate while you are imposing the stress. Moving ahead, another thing which is the validations. So the validations in which if you see the values, the validation is different in the both the conditions. The similarly way the amplifications, floats and the melt curve seems to be a different. If you see that the different genes which regulating while you are imposing the drought stress, in the gene 16, in the stress conditions in the wild cultivar, it is upregulated, and the similarly way in the stress conditions in the 22, it is also upregulated. The same way the gene 82 also regulated in the revival conditions in the 14.496. So I can say that in the experiment, the wild cultivars and the cultivated varieties in the experiment can be says that this can be a key features in which you will get the certain genes maybe regulate while you are imposing the stress. So it can be concluded from the above studies that during the drought stress, the drought tolerance wild types exhibited a various physiological, biochemicals, and the molecular changes to survive the water deficiency, while the GJG22 exhibit the minor variations and so is susceptible to drought conditions. I can add on this to say this upon the rewatering the plants, the recovered displaying the reading similar to the pre-stress control conditions. The QRT-PCR analysis correlates with the with the physiological and biochemical data which is reflected. Apart from this, I can say this gene arc, which having the different C-terminal regions, a variable C-terminal regions, may be responsible for the improving the chart tolerance by regulating the opening and the closing of the stomata and improving the water use efficiencies and the many other such mechanisms. Thank you very much, guys, for listening to this.